can we get started? All right. Anything before we go on? Okay, what we're going to talk about now are the nature of the interaction between antigens and antibodies. And then we'll spend some time talking about some of the tests that employ antigens and antibodies to measure things for you. And the tests that we'll go over are things that would be commonly done in a clinical lab. Now, some of these, obviously, it depends. Which test is used depends upon the throughput in the particular lab that you'll be associated with. Some of them, very large labs, will have more sophisticated ways of doing this. But what we want you to get out of this is sort of the principle behind these types of tests and that use antigen-antibody reactions to measure things for you. Okay, let's talk first about the nature of these interactions. Obviously, our concept of the antigen-antibody reaction is a lock and key. We've been saying that the hypervariable regions of the heavy and light chain come together to create a pocket into which the antigen is going to insert. All right. Now, all of the interactions that occur between the antigen and the antibody are non-covalent interactions. Right? You do not see in antigen-antibody reactions any covalence. So you're not going to see carbon-carbon bonds. You're not going to see disulfide bonds, anything like that. They're all non-covalent interactions. So you're going to see things like hydrogen bonds, electrostatic bonds, van der Waals forces, hydrophobic bonds. Now, in any individual interaction between antigen and antibody, the proportion of these may vary. So, for example, if you've got a highly charged antigen, it's more likely that the interactions are going to be primarily electrostatic interactions in that case, all right? If you have something that's very hydrophobic, then you're not going to have electrostatic bonds there. You're going to have more hydrophobic bonds. So these will vary depending upon the particular interaction that's going on. But in all cases, they're non-covalent. Now, there are some consequences of the fact that they're non-covalent, and one consequence then is that in order for an antigen, since non-covalent bonds are weak bonds, in order for an antigen and antibody to be able to stay together, there's going to have to be multiple interactions between the antigen and the antibody. And what's depicted here is actually, this is just the Fab fragment. You're seeing in the ribbon diagram, you're seeing the Fab fragment. One of these is the heavy chain, one of these is the light chain, coming together to form the hypervariable regions are positioned to bind the antigen. And the antigen is here in this space-filled model. And you can see that there's multiple places where the antigen makes contact with the antibody molecule. And that's because these are non-covalent interactions. If there was just one or two of these, they would never be able to stay together because these bonds, non-covalent bonds, are weak bonds. The other consequence of the fact that these are weak bonds are non-covalent bonds is that all antigen-antibody reactions are reversible, because if that bond can form, that bond can dissociate, and the complexes can dissociate. So antigen-antibody complexes, by their nature, are reversible. Now, there are two terms that are used to describe the strength of binding between an antigen and an antibody. Those terms are affinity and avidity, and we'll discuss both of them. We'll start with affinity. Affinity is the strength of the reaction between a single antigenic determinant and a single antibody combining site. So the strength of that binding is going to be the affinity. And so let's take a look at a situation. We immunize with an antigen and we produce an antibody. The arrows pointing towards each other are to imply attractive forces. So there's a lot of attractive forces holding this antigen into the antibody combining site. There may be another antigen that has as a epitope that is structurally similar, but certainly a little bit different. So there may be some attractive forces between the antigen and the antibody, but in addition, the red arrows pointing away from each other are to imply repulsive forces. There are some repulsive forces. So this would have bind with a higher affinity than, this antigen would bind to the antibody with a higher affinity than this antigen. And the affinity is nothing else but just the sum of all those attractive minus any of those repulsive forces. That gives you the affinity. It's the strength of the binding between an individual epitope and a single combining site. Now you can actually put a number to that. We can actually calculate the affinity. And the affinity is nothing else but the equilibrium constant that describes a reversible reaction. So if we go back to your chemistry now, if you remember antigen, if you take substrates reacting in a reversible manner to form a product, you can write an equilibrium constant that is equal to then the molar concentration of the product divided by the product of the molar concentrations of the reactants. All right? Just simply applying the law of mass action. This number is the equilibrium constant, is the affinity. And in antigen-antibody reactions, the affinity is very, very high, which is just another way of saying is that this equilibrium favors the production of product. All right? So these are pretty high. So this is the equilibrium is shifted in this direction to a large extent. It's a very high affinity. All right. The other term that's used to describe the strength of binding between antigens and antibodies is avidity. So avidity is the overall strength of binding between an antigen with multiple antigenic determinants and multivalent antibodies. Affinity, we dealt with just a single reaction going on, a single combining site reacting with a single epitope. But that's not what we have in the real world. In the real world, antibodies are at least divalent, right? At the minimum, they're divalent. IgM can be pentavalent. So the antibodies are at least divalent. And most antigens, with the exception of haptens, are multivalent. Most antigens are going to have more than one epitope. Haptens are the exceptions. Haptens are small. They only have a valence of one, okay? So in the real world, when antigens and antibody react, we have multiple epitopes reacting with multiple combining sites. And so we can calculate a number for the affinity, or the equilibrium constant. But what we're really measuring is not affinity. What we're measuring is avidity. And let me illustrate. So here again is affinity. We have a single epitope reacting with a single arm of the antibody molecule. We measure the equilibrium constant. We come up with a number. That's a measure of affinity. All right? That's affinity. But notice what happens when you take into consideration valence of both antigen and antibody. Here we have a divalent antigen reacting with a divalent antibody. So we have an immune complex. The only way that this immune complex dissociates back into free antigen and free antibody is if both arms of the antibody molecule release simultaneously. That's the only way you can generate free antigen and free antibody. If one arm dissociates, you still have an immune complex because the other arm is still attached. So it's still going to be counted on the product side of that equation. So the only way that you dissociate that is if they both dissociate simultaneously. So we can calculate what we think is the quotes affinity. Or we can calculate the equilibrium constant, and we think that that's affinity. But it's not affinity. It's much higher. The number we get is much higher. The affinity of each of these arms interacting individually is 10 to the 4. The overall affinity of binding is 10 to the 6. So the overall is the avidity. It takes into consideration that we're dealing with multivalent antigens and multivalent antibodies. Look what happens with something like IgM. The only way this dissociates is if every arm of the immunoglobulin dissociates at the same instant. All right? If any one of these, even if 5 of these dissociate and one of them doesn't, it's still counted as an immune complex, and so it's still counted on the product side of the equation. All right? And so we come up with a number that we can measure. But that's not a measure of the affinity. That's the measure of the avidity. It's the overall strength of binding because of valence. Each arm here, each individual reaction is got an affinity of 10 to the 4. The overall strength of the binding is much, much higher. And that's the avidity. So anytime you have valence into this thing, you're going to really, when you make a measurement of the strength of the binding, you're really measuring avidity. Absolutely, because any individual molecule, remember, has got all identical heavy chains and identical light chains. So in this IgM, every one of these is the same epitope. Now, what you have, of course, also in the real life situation, you have multiple antibodies directed against multiple different specificities. But they can, they can still. So even if you had two antibodies, let me see if I can. If you had a situation like this, here's your antigen with one kind of epitope versus another kind of epitope, and then we have antibodies binding here. We have one antibody, say, binding to this one, and a different antibody binding to this one. You still, even if this one dissociates, it's still an immune complex because this one's still there. Right? So, all right. All right. Now, again, there are two other terms, too, that when you talk about antigen-antibody reactions that are important to keep into consideration. And that is specificity and cross-reactivity. So what is specificity? Specificity is the ability of an individual antibody combining site to react with only one antigenic determinant. If a particular antibody combining site only recognizes one epitope and will not bind absolutely anything else, that is said to be specific. All right? This term, however, is also used in another context. It's used to describe the ability of a population of antibody molecules to react with only one antigen. And we've used it in this context before. Remember when we did that little exercise with isotypes and we took that human IgM, we injected it into a rabbit, we got an antibody, we got an antiserum, then we absorbed that antiserum with IgG and IgD and IgE and all those things. We ended up with a serum that was specific for IgM. It only reacted with IgM. It didn't react with the others, all right? So that we can use the term specificity in that sense as well. So we had a population of antibody molecules, but they only reacted with one antigen. It didn't react with all the other immunoglobulins, okay? That's an example of specificity. So we would say that that antiserum was specific for IgM. The other term is cross-reactivity, which is just the opposite, is the ability of an antibody combining site to react with more than one antigenic determinant. So if the antibody combining site can recognize more than one antigenic determinant, that is cross-reactivity. The other term that's used for that we just used in the last hour was multispecificity, is another way of saying it's cross-reactivity or multispecificity. This term, however, is also used in this other context as the ability of a population of antibody molecules to react with more than one antigen. When we did that little exercise and injected the IgM, the first antiserum that we got out reacted with everything. So we would say that that antiserum cross-reacted with IgG and IgD and IgE and IgA. So that was a cross-reacting population of antibody molecules. All right, so those terms are used in that way. Now, obviously, if you're going to use antigen-antibody complexes to measure things for you, then you have to be confident of the specificity of your antibodies. If the antibody is not specific for what you think it is, then you're not measuring what you think you're measuring. So, for example, if we had taken that that non-adsorbed antibody that we made by just injecting the human IgM into that animal, and we used that to try to measure IgM levels, for example, in a patient, we would be in big trouble because we're not measuring IgM. We're measuring everything because it was cross-reacting with everything, all right? So in all these cases, when you deal with antigen-antibody reactions, cross-reactivities, it's very important that you know something about the specificity or cross-reactivity of your antibody molecules. All right. So how do cross-reactions arise? They arise in two ways. Here's the situation. We immunize with antigen A, so we've got an epitope that this antibody is directed against. So cross-reactions, these antibodies can cross-react in one of two ways. One way is if a different antigen shares an epitope. It may be that antigen B has the exact same epitope as antigen A. Obviously, then these antibodies to A will bind to that antigen, so this would cross-react. The other way cross-reactions arise is if there is the cross-reacting antigen has an epitope that is similar to the immunizing epitope. All right. So here we have an epitope that is structurally similar, and it might be able to fit into the binding site. Now, this one will probably bind with a lower affinity than this one. But as long as the affinity is high enough, it'll still bind. Yes? Can you have multiple epitopes on the same antigen? Sure. 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 Yeah, in other words, you're saying, could this antigen also have something like this, like antigen C? Sure. Absolutely. Okay? All right. Before we get into the tests for the actual tests that are used that employ antigen antibody reactions, I just want to talk about a couple things that influence the ease with which you can detect these complexes. I mean, the only way that you know that an antigen and an antibody have reacted is if you have some way of detecting that complex. All right? So that's the whole trick in this. You have to have a way of recognizing the immune complex, or identifying that and isolating in some way, getting that immune complex. All right? And there are several things that can influence how easy it is to measure or detect those complexes. One thing is obviously the affinity. The higher the affinity of the antigen for the antibody, obviously the stronger the binding is going to be, and the easier you're going to be able to do a lot more things to try to identify those antigen-antibody complexes. So you'll be able to do a lot more drastic techniques in order to try to recognize them. So affinity obviously plays a role. And of course, avidity plays a role too, because if affinity is going, the higher the avidity, the tighter it's going to stay together, the more you're going to be able to do to try to detect those complexes. Now, what's less obvious is that the antigen-antibody ratio is also very important. And let's illustrate why. Suppose you have a situation. We have the antigen's going to be here with these blue balls on the end, okay? It's a trivalent antigen, okay? And we have a divalent antibody. Look what happens if you go in antibody excess. In a situation where you have an excess of antibody over antigen, the largest immune, or the predominant form of the immune complex is going to look like this. You're going to have one antigen molecule associated with 3 antibody molecules. That's going to be the predominant form. Now, there may be some that are a little bit bigger than that, but that's the predominant form of the immune complex. Now, it is immune complex, and it's there. The problem is that it's very small. And how are you going to try to detect something that is essentially only 4 molecules, 3 antibodies and 1 antigen? So it's there, but it's going to be more difficult to detect. If you go to the other extreme and put antigen in excess, then the predominant form of the antibody, or the complexes, are going to look like this, where you have 2 antigen molecules cross-linked by an antibody molecule. Again, it's an immune complex, but it's very small. You're going to have difficulties trying to detect those things, or you're going to have to come up with some ways to do that if you want to work on the antigen excess. But when you get the right ratio of antigen to antibody, what happens is that essentially every antigen molecule is cross-linked to another antigen by at least 1 antibody molecule. And you get what is referred to as lattice formation. You get this big cross-linking of the antigens and antibodies. And these complexes become to be so big that they actually can become visible even to the naked eye sometimes. All right? So these obviously are a lot easier to detect than either of these. So you've got to, when you deal with antigen-antibody reactions, the ratio of antigen to antibody is going to become important. The point at which essentially all the antigen and all the antibody are in complex, and then you have no free antigen for antibody, is called the equivalence point. And so ideally, you try to get effective lattice formation. You try to get at or as close as you can get to that equivalence point, because that's when you're going to have the most cross-linking of antigen and antibody. So ratio is important. Another thing that is important is the physical form of the antigen. This really affects how you read, what the readout is, more than the ease with which you can read it. If the antigen is particulate, as if it's a particle like a red blood cell or a bacterium, the readout is going to be agglutination. You're going to just look to see if they clump. All right. If the antigen is soluble, then the readout is going to be precipitation. What happens when these antigens are soluble is these complexes get so big they actually precipitate out, and you can just look or measure the precipitate. All right. All right. A couple things to keep in mind. That all tests based on antigen-antibody reactions are either going to have to depend upon lattice formation. You're going to have to find the right ratio of antigen to antibody. Or else they're going to have ways to detect those small immune complexes. Those are your only choices. Get lattice formation or try to find ways to detect those small immune complexes. The other point I want to make is that any test for antigen-antibody reactions can be used to detect either the antigen or the antibody. So if you put in a standard amount of antibody, you're measuring antigen. If you put in a standard amount of antigen, you're measuring antibody. All right? So they can be done either way. Now, certain tests are more commonly used for measuring antibody, and some tests are more commonly used for measuring antigen. And that's why I'm going to describe the tests as we go through them. But keep in mind that you really can you tweak the test so that you can measure either antigen or antibody by any of these tests. All right. The first group of tests are called the agglutination tests. These will depend on lattice formation. The first test is simply called an agglutination. Sometimes you see them referred to as hemagglutination test, and that's because very often the particle that is used in this test is a red blood cell, and so they're called hemagglutination tests. And this is nothing else but a test that has its endpoint looking for agglutination. Now, you may come across these terms, agglutinin or hemagglutinin. This refers to the antibody that is doing the agglutination. And it's unfortunate. It sort of gives you the impression that this is some sort of special antibody. It's not. It's a functional term. A functional term that's an antibody that causes agglutination is referred to as an agglutinin. But there's nothing special about that antibody. Okay. The test can be done just simple qualitative tests. You want to detect either antigen or antibody. What do? You simply take, if you're trying to detect antigen, you take a known amount of antibody, you mix it with antigen, and you look for agglutination. All right? Or if you want to really look for antibody, you take a fixed set amount of antigen, mix it with the unknown, and you look for it to see if the antibody's there. This is what's common. If you've ever done blood typing, if you've been blood typed, this is what they do. They take a drop of your blood, they mix it with antibodies to the A blood group antigen, and they look on a slide to see if it agglutinates. If it does, then you've got blood group A. Then they take another drop of blood, mix it with antibodies to the B blood group antigen. If it agglutinates, you're type B. If you don't agglutinate in either of those tests, you're type O, and if you agglutinate in both, you're AB. It's that simple. A very qualitative type test. But you can also do these tests in a more quantitative way. And this is most commonly done for looking for antibodies. So, for example, you might send a patient sample down that you want to look for antibodies to a particular bacterium, for example. And so, you want to have some quantitation as to what the antibody levels are. Then we'll do a quantitative agglutination test. It's most commonly done in one of these plates that have 96 well called a microtiter plate. And what they do in the lab is they'll go ahead and they'll take your patient's serum, so patient 1, and they'll make a series of serial two-fold dilutions of your patient's serum, all the way out to usually about the 10th well. The 11th and 12th well are set there for controls. One is something that they know is going to agglutinate the particles. The other one, something that will not agglutinate the particles, okay? All right, so what's going to happen? So you dilute out your patient's sample. Then they come back with the particulate antigen. So in your case, if you're looking for a bacterial infection, they'll put the bacteria in the well, okay? And they'll put a standard amount of bacteria in all these wells. Well, what's going to happen? If there's antigen there, antibodies there to the bacteria, they're going to cross-link the bacteria and agglutinate them. When they reach the point where they've got an effective lattice formation, these complexes are going to get very big. And they actually settle out. And they'll fall to the bottom of the well, and they'll cover the whole bottom of the well. If there's no antibodies there, the bacteria will settle out eventually, but they settle out right in a small button at the bottom. So a negative, when there's nothing agglutinating it, you just get this little button. A positive, when you get agglutination, you get it covered over the whole well, and you can just look at it and see. And so the way the quantitation is done is to take the highest of the titer, which is the measure of the concentration of the antibody, is nothing else but the reciprocal of the highest dilution that gives you visible agglutination. So here the highest dilution that gives agglutination is 64. The titer would be reported out as 64. So you would get a report that the antibody titer is 64. Okay? This one has got a titer of 8. All right? Now, this is semi-quantitative in the sense that you don't know exactly how much antibody is there. You do know that this person has got 8 times more antibody than this person. So relatively speaking, it's a relative quantitation. Okay? Here's an interesting case. Look at this patient here. There's no agglutination here, followed by agglutination, and then it disappears again. All right? This is called a prozone, where you see no agglutination prior to, and as you dilute it, you see agglutination. What do you think is responsible for the prozone? Exactly. It's antibody excess. Antibody excess, so you're not getting lattice formation. You don't get agglutination. But then as you dilute it out, you finally reach a point where you're getting good lattice formation, and then you finally dilute it out. So the titer of this individual would be 512. You ignore this up here because you know that that's due to too much antibody. The titer is 512. Okay? All right, so where do you use these things? Well, in blood typing, the qualitative test is used in blood typing. For bacterial infections, the quantitative or semi-quantitative test is used a lot. Now, when you do this test, though, for bacterial infections, that's all you know is that there's antibody there, and you get a relative measure of how much antibody is there. But what it doesn't tell you whether or not that antibody is from a current infection or a past infection. It just says that there's antibody there. So what you have to do, actually, in order to determine whether or not if your patient is undergoing a current infection with the bacterium that you suspect may be going on, is you have to run a paired set of samples. You take a sample early on when you see the patient, then you take a sample by maybe in about another week or so. And the lab has to run both samples. And what you're looking for, if it is a current infection, what will happen is the antibody titer will rise because the immune system is being stimulated. If it is a past infection, the antibody titer will either stay the same or might drop a little bit. So you really have to send to the lab a paired sample, all right, for them to do this. And also, in this, you're also going to have to look for a 4-fold rise in titer before you can conclusively say or confidently say that this is a current infection. Now, why a 4-fold rise in titer? In the previous slide, I made it nice and simple. It was quite clear. This is the last agglutination. The next one, there's no agglutination. Well, in the real world, it's not quite that simple. What you see oftentimes are things like this. So what do you call that? Is that agglutination or is it not agglutination? In other words, what is the titer that's going to be reported out to you? The tech that reads this. Is he or she going to report out a titer of 64, or are you going to get a report out of a titer of 128? There's some subjectivity in the tech that reads this, all right? And so what you do is usually by the next well, it's going to be perfectly clear that there's no agglutination. So the only way you can be confident that this is a current infection is if you see a 4-fold rise in titer, which is essentially 2 wells, because you're making serial 2-fold dilutions. So you always are looking for at least a 4-fold rise in titer when you send your set of paired samples. All right. These are very easy tests to do. They don't require a lot of sophisticated equipment. That's all you really require is a little microtiter plate, some pipettes, and it's very simple to do. They're only semi-quantitative at best because you get relative concentrations. Now, there's another test which is essentially the same as what we've just described, but it's called passive agglutination. The way the agglutination test, by definition, has got to have a particular antigen, right? Because agglutination is the readout. Well, what happens if you're interested in looking, at a soluble antigen, and you want to use this test because they're so simple to do? So then what they do is a passive agglutination. And passive agglutination is nothing else, but you take the soluble antigen and coat it on some particle. Typically, they're coated on erythrocytes or on latex beads, anything like this. They chemically coat the antigen, the soluble antigen on there, and then you just do the same thing. So here we have our particle now coated with the soluble antigen in blue, and then you simply set up your test, either a qualitative test or a quantitative. You can make dilutions as well to measure antibodies to a soluble antigen. So it's the exact same test. The only difference is that they chemically coated it onto a particle. Now, there are two special tests that are used a lot in the clinical labs, and those are called the Coombs tests. There's actually two of them, a direct and an indirect Coombs test. And the Coombs test actually was developed to detect something called an incomplete antibody. And this is really an unfortunate term because it sort of gives you the impression that there's something wrong with it, that this is only a piece of an antibody molecule. That's not the case. An incomplete antibody was an antibody that could not agglutinate particles. All right? Even if you achieved what should be the appropriate ratio of antigen to antibody, in order to agglutinate particles, like particularly red blood cells, which is where it was developed, the red blood cells are very highly negatively charged. You really got to overcome the high repulsive forces of erythrocytes for each other. So sometimes the antibodies just can't do that. When they bind, and they just can't cross-link enough in order to bring big agglutinates because of the repulsive forces that are naturally present on the particles. So those antibodies were called incomplete antibodies because they couldn't agglutinate. And again, it doesn't mean that they're pieces of antibodies. They're complete antibodies. It's just that they can't agglutinate. All right. So the Coombs tests were developed to try to deal with this problem of incomplete antibodies. So the direct Coombs test, what it does is it is a test that detects antibodies on erythrocytes. So if you suspect that your patient has got erythrocytes that are covered with antibodies, and they're not agglutinating the erythrocytes in any way, you want to test to see whether or not there's antibody on that patient's erythrocytes, you do the direct Coombs test. So here we have a patient that we suspect may have antibodies on his or her red blood cells. What you do in the direct Coombs test is you mix it with the Coombs reagent. And the Coombs reagent is nothing else but an antiglobulin. It's an antibody directed against antibody. And it can be an anti-isotypic antibody, for example. All right? So it's an antibody directed against these human antibodies that are on your patient's serum. And what you do in the direct Coombs is you let this antibody do the cross-linking and agglutination for you. So you mix the patient's erythrocytes with the Coombs reagent and you look for agglutination. A positive test, agglutination, a positive agglutination in the direct test means that your patient's sample did in fact have antibodies on the surface. A negative direct Coombs test means that there were no antibodies on your patient's erythrocytes. Okay? That's the direct test. The indirect test asks a different question, or attempts to answer a different question. It's used to detect whether or not your patient's serum has antibodies in him, which will react with an erythrocyte, or any particle for that matter, but typically for erythrocyte, which will react with erythrocytes. And you're concerned that this might be antibodies of this incomplete type. So you're asking whether or not the patient's serum has antibodies to the erythrocytes. All right? This is the two-step test. The first thing you do is you take the patient's serum that you're now interested in testing for antibodies. You mix it with the target cells that you think the antibodies are directed against. All right? They'll react. Of course, if they agglutinate, there's no problem. You know that they're there. But what happens if they don't agglutinate? If they don't agglutinate, you don't know, did I not get the ratio right? Am I dealing with incomplete antibodies? You don't know what the situation is. So what you do in the indirect Coombs test is the second step is to take these cells, and obviously what you do is after you let this reaction go, there's a washing step in here. You wash out all this other stuff, and you get these, and now you add the Coombs reagent to the washed cells. If there's antibody there on the cells, then it'll agglutinate. So a positive indirect Coombs test means that your patient's sample or serum did in fact have antibodies in them to those erythrocytes. Yes.
Why couldn't you go straight
To here to here? Well, then yes, that reaction would work, but now you're dealing with a soluble antigen and the antibody. You're not going to see agglutination. These tests are based on agglutination. I mean, yes, there might be ways of doing that, of course, but this is just a simple way of, because these tests are so simple to do. These agglutination tests. I mean, they're inexpensive and very simple to do. That's what a clinical lab has got to have. Easy things to do. Okay. All right, so where we use this? These tests were actually developed to detect antibodies to the Rh factor. Antibodies to the Rh factor are notorious for being incomplete antibodies. In other words, they don't agglutinate erythrocytes. All right, so the Coombs test was actually developed to deal with this situation. So, for example, if you suspect, if there's a baby born that you suspect might be suffering from erythroblastosis fetalis because there's an Rh incompatibility between the child and the mother, which test would you do on the child? Which Coombs test would you do, the direct or the indirect? The direct. You want to know whether or not that child's erythrocytes are coated with antibodies. What might you test, what might you want to do on the mother, even probably before she delivers? The indirect, because you want to know whether or not she has antibodies to the Rh factor. All right, so you would do the indirect Coombs test on the mother. Okay? Although it was developed for Rh testing, in fact, these tests can be used for other things. These are commonly used for detecting autoimmune hemolytic anemia. If you're dealing with a patient that has an autoimmune, and they start making antibodies against his or her own erythrocytes, what are you going to do? Which test? Direct. You're going to take some of the cells and you're just going to do a direct Coombs test to see whether or not the erythrocytes are coated with antibody. Okay? So there are other applications besides the Rh, but that's what these Coombs tests were actually developed for. All right, hemagglutination inhibition. This is a test based on the inhibition of agglutination due to competition with the soluble antigen, and it is also a two-step test. We take our passively coated erythrocytes, mix it, and prior to the actual doing the test, you determine what's the best ratio between antigen to antibody to get a good agglutination. All right. Now the test is actually this. You take this predetermined amount of antibody, the predetermined amount of antigen on your bead, and you mix it with the patient sample that you're interested in finding out whether or not that patient sample has the antigen and how much. All right. All right, so what's going to happen? If the patient sample does have antigen, then that antigen is going to compete with the antigen that's bound to the particle for binding to these antibodies. And you will block the agglutination. The antibodies will be tied up binding to the soluble antigen and won't be able to cross-link the erythrocyte. So a positive hemagglutination inhibition test means a lack of agglutination. All right? So this is a positive test, which means that your patient sample did in fact have antibody, the antigen in question. Okay? All right. Again, so this is used for measuring soluble antigens. And these are just easy tests. All right. The next group of tests are the precipitation tests. These also rely on lattice formation. First one is called a radial immunodiffusion. Now, these precipitation tests, while they can be done in solution, are typically done in gels, all right? And you look for the precipitation right in the gel. It just makes it's more convenient to do that. So the tests I'm going to describe are gel-based tests. This is the radial immunodiffusion or Mancini test. What you do is you take a slide and you pour a gel onto the slide, and you incorporate into the gel the antibody. All right, so your antibody is incorporated into the gel. Then you poke a couple holes in this gel, and into that gel you put the antigen to that antibody in increasing concentration. So these are known concentrations of antigen that you're putting in the wells. Well, what's going to happen? That antigen is going to start diffusing into the gel. course, immediately, right next to the wall of the well, you're going to be at a high antigen concentration. So you're going to be in antigen excess. There won't be any precipitate, right? But as the antigen continues to diffuse into the gel, eventually you reach a spot, a point, where the ratio of antigen to antibody is appropriate, and a precipitation will occur. And of course, since this antigen is diffusing out in all directions, you'll get a ring of precipitation. You'll see a precipitation ring. All right? Now, this test is actually quantitative because you can measure the diameter of the ring of precipitation is related to the concentration. The higher the concentration of the antigen, the more it's going to have to diffuse before you reach that point of equivalence. And so the diameter of this ring of precipitation is proportional to the antigen concentration. So you can then go ahead and set up a standard curve where you can relate antigen concentration to the diameter of this ring. It's actually, the linear relationship is to the square of the diameter. So you take the diameter, square it, and you make a plot of antigen concentration versus diameter. Then you take another hole, you poke it in that agar, and put your patient sample in. Measure the ring of precipitation, and come to your graph and just read off what the antigen concentration is. So it's a quantitative type test. This is what it can maybe use in your hospital labs to measure immunoglobulin levels. What they do is they put antibody, for example, to IgG in the well, and then you take your patient's serum and you put it in the well, and you generate a standard curve, and they'll tell you what your antigen concentration is, what your IgG level is, or what your IgM level. So this is commonly used. Now, there are other ways of doing it, and again, whether or not your lab uses this kind of a test really depends on its volume. If it's got a big volume, there are other ways to do it besides these things. But if the volume is fairly small, this is a very simple way to do the thing. Again, doesn't require a lot of sophisticated equipment. That's all you have to do is have a ruler and a magnifying glass to measure the diameter of that ring of precipitation. Yes? You actually, what happens, obviously this is a cartoon of it, but what you see is actually a white ring. Because the immune complexes get so large, they actually precipitate out right in the gel. And you see a white ring around the well, and you just measure the diameter of that ring, that circle. Okay, immunoelectrophoresis. This is a technique that's used when you're analyzing complex mixtures of antigens, like human serum, for example, which got a lot of different proteins in it. What you do is you pour a gel on a plate, on a slide or a plate, and then you poke one hole in it, and you put antigen. So you put, let's say you're doing this for analyzing human serum. You would put fill that well with human serum. And then you'd apply an electric current, and you'd use the electric current to separate the proteins based on their charge. Okay? So you'll end up, after the electrophoresis, the proteins will be separated based on their net charge. Then what you do is you cut a trough in this gel, and you fill it with antibodies. So in this case, we said if this was human serum that we put in here, we would put antibodies made against whole human serum. And what's going to happen? The antibody is going to be diffusing into the gel and encounter the antigen. Where you reach the point of equivalence, a precipitation will occur. And these will be arcs of precipitation. They'll look like this. The skilled technician will be able to look at this and be able to identify this arc is IgG, this arc is IgA, this arc is IgM. So, and it's really, I've been trying to get a good picture of one of these things. There's actually over 30 different arcs in there. But the skilled technician will look at it and say, this is what this is this. Like, for example, you know that this one is IgG because IgG travels the furthest. We know that. So they'll look at this arc and then say that's IgG, and they'll this one IgM and IgDA. And so they can actually identify all the serum components by looking at these arcs, okay? So where would you use this for? Well, one of the ways it's used to help diagnose immunodeficiencies. If you have a patient that has an IgG deficiency, that arc's going to be missing. Or if it's not missing, it may be reduced in its thickness. So you can get some idea of concentration by the thickness of these arcs. But So in deficiencies, they're either a little bit thinner or they're gone completely. And so they are, by a very simple test, they can look at it and say, this person doesn't have any IgG. Okay? All right. One last precipitation test, countercurrent electrophoresis. This has rather limited use, but it's when it can be used, it's very helpful. What you do in the countercurrent electrophoresis is you pour your gel on a slide, you poke a hole for antigen and poke a well for antibody. You fill this one with antigen and antibody, and then you apply the electric current. And you electrophorese them towards each other. You don't wait for them to diffuse. You actually electrophorese them. Now, obviously, this test has more limited usefulness because this only works if the antigen and antibodies have opposite charges so that they move in opposite directions in the electrical field. The advantage of this is that it is very rapid. Within a half hour, you can see your line of precipitation occurring here. You do any of those other tests, like the Mancini, the radioimmunity diffusion, you're talking about 24 hours to let that diffuse into there and see your band of precipitation. This can be done in 30 minutes. And so there are some tests out there, for example, that use this technique. When in instances where infections, you need to know right away. There's, for example, cryptococcal neoformans meningitis. You need to know immediately whether or not that's what your patient's dealing with, because if you don't treat, the patient's going to die. All right? And so there's a countercurrent electrophoresis test that measures for a cryptococcal antigen that uses this technique because it's very quick. Again, it's qualitative. You're not quantitative. That's all you want to know is the antigen present? And this will tell you. And it'll tell you in 30 minutes rather than a day. Okay, let's stop here and we'll pick up at this point on Monday.